Well, good evening and welcome everyone. My name is Susan Fernbach. I'm the co-director for the Office of Community Engagement and Diversity for the Department of Molecular and Human Genetics at Baylor College of Medicine. And we welcome each of you. Tonight is the 16th anniversary of the Evenings with Genetics program, which is a free series open to the community. During the month of February, we are recognizing and celebrating Black History Month and the second annual Evenings with Genetics webinar on race and genetics, perspectives on precision medicine. Due to the ongoing social issues in our country, we felt there remains a need to address the old concepts of genetics and race, to discuss some of the causes of health inequities or inequalities, and to enable us to improve access and care for people in all of our communities, and especially in our communities of color. This webinar is co-sponsored by the Department of Molecular and Human Genetics and the Baylor Office of Diversity, Equity, and Inclusion. Tonight we'll offer simultaneous interpretation to Spanish, and please see the interpretation box below. During the webinar, you can enter questions in the Q&A box at the bottom of your screen. Dr. Deborah Murray, co-director for the Office of Community Engagement and Diversity at Baylor College of Medicine, will moderate the discussion and questions. We have a wonderful evening planned with 3 speakers, 2 of whom are on East Coast time, so we are greatly appreciative of all 3 for speaking tonight. I will introduce each of them briefly, and please see the link to their full biographical information on the Evenings with Genetics webpage. So our first speaker tonight is Mr. Vence Bonham, the Acting Deputy Director for the National Human Genome Research Institute. Mr. Bonham provides leadership for the Institute's health equity and workforce diversity programs and works in partnership across the NIH to promote the mission of the Institute. He received his Bachelor of Arts degree from James Madison College at Michigan State University and his Juris Doctor degree from the Moritz College of Law at Ohio State University. His research focuses on the social implications of new genomic knowledge, particularly in communities of color. He studies how genomics influences the use of the constructs of race and ethnicity in biomedical research and clinical care, and the role of genomics in either making health inequities worse or improving them. The Bonham Group also studies sickle cell disease, a condition that will be impacted by emerging curative genomic technologies, but has faced significant health inequality both in the US and globally. So please give a warm welcome to Mr. Vence Bonham.
So thank you.
Take it away, Mr. Bonham.
Thank you, and thank you for the invitation this evening. So let's see if I can get my slides up here. Okay, are my slides up? Well, I first just want to again thank you for the invitation to come and be part of this series that I really think is really great, because it's not just for researchers and students, but it's also for the community, and that an opportunity for me this evening to share some things that are going on at the National Human Genome Research Institute to the public and to the community in Houston, Texas, and the Baylor community. This evening, what I want to do with my time is to take on 2 different issues and to talk about them. One is health equity, and then the second is really focused on the issues of what does race mean with regards to genetics and genomics. But I start by giving you greetings from the National Institutes of Health. So the National Institutes of Health is made up of 27 different institutes, centers, and offices. It's located in Bethesda, Maryland, and it is the engine of biomedical research in our country. We fund from the federal government the vast majority of all biomedical research, both at Baylor College of Medicine, but across the country and across the globe. And we want to bring you greetings from the National Institutes of Health, because our work is really to seek to benefit all communities across our country and across the world. But what I want to do with my time tonight is to talk a little bit about precision and genomic medicine. And I'm really going to frame it around genomic medicine of how we use our knowledge with regards to our genomes to understand how to provide better care, individualized care for individual patients in need. And I like this slide that I received from my director, Dr. Eric Green, and I bring greetings also from Eric about the future, that the exit ramp is here now, that we are truly having the reality of our ability to use genomic medicine and precision medicine to improve the health of populations. And it's exciting all the things that are happening today within the field of genomics. It is a very exciting time. And the potential benefit both for rare disease and common disease is great. It's happening now. It's not the future. We are now there where we are beginning to have the implementation of genomic medicine to improve health of populations. But we have challenges, and that's why I want to talk about health equity in genomics. And the framing of this issue about whether this new knowledge, new technologies will actually benefit everyone. And I use this definition of health equity from the Robert Wood Johnson Foundation, that health equity means that everyone has a fair chance on, let's see if I can move this, a fair and just opportunity to be as healthy as possible. And so when we think about health equity in the face of genomics, what do we need to think about to make sure that everyone can benefit? So let me just take a minute and talk about what I mean when I think about genomic and precision medicine and the questions of who will benefit. So when we think about genomic and precision medicine, we're talking about diagnostics. Can we determine better ways to diagnose diseases, and particularly those rare diseases that genomic information may be the way to truly understand what may be the disease impacting an individual patient? The issue of pharmacogenomics. How can we tailor and provide the best drug, the best medication for an individual based on their genomic makeup, and to make sure that they don't receive drugs that could adversely impact them and cause them harm? And so how do we make sure that we're creating precision, precise healthcare for individual patients? The issue of risk assessment, to understand risk. What is your risk of getting specific types of cancer? What is your risk of heart disease? How can we provide you information early so you can change your behaviors to actually reduce your risk? And this too is a part of genomic and precision medicine. But the last part, which is also very exciting, is the development of new drugs and opportunities to do gene therapy, gene editing, gene addition, modifying a genome to actually address someone with the disease. One of the areas that I am involved in research, as stated, is sickle cell disease. And it's a very exciting time with regards to sickle cell disease and gene therapy. This ability to actually make a change, to have a curative therapy for a disease with such a terrible burden. But the issue that I want to focus on is that diverse communities face barriers in accessing genomic and precision medicine. And when I think about diverse communities, I'm thinking about those communities that have not been part of research, have been underserved historically. How do we make sure they too benefit? So I want to share with you a study that was conducted in 2016. And this study was by Dr. Alice Popejoy and Dr. Stephanie Malia Fullerton at the University of Washington, where they replicated a study that was done in 2009. And in 2009, there was a study conducted of what's called genome-wide association studies. These are studies to understand and identify gene variants in individuals and in populations. And in 2009, it was shown that of all of the studies that had been conducted, 96% of all of the studies have been done on a European ancestral population. Now, let me just stop for a minute. When you look globally, European ancestral populations make up somewhere around 10% of our global population. But the research was conducted was only in 96% of us in European ancestral populations. So when Dr. Popejoy did her study, they replicated the work that was done in 2009 and 2016, and they showed that there had been change, that the percentage of European ancestral populations in the study had reduced to 81%. But they also showed a couple of things about that change that occurred, that the increase in the non-European populations or samples and participants in the studies, that 78% of that increase had come from Asian ancestral populations, where there had been major genomic research institutes developed in China and other parts of East Asia. And that was the growth that we were seeing with regards to the diversity of the populations. And when you think about the United States and you think about the populations in the United States, still less than 4% were from diverse populations, African American and Black Americans, Latinx and Hispanic Americans, Indigenous, American Indian, Alaskan Native communities were all missing from the research. And so what does that mean when those populations are not part of the research? What it means is that we can't study to understand variants in individuals' genomes that may impact disease and may be benign and have no impact. And so we need that information to understand the global diversity in our populations and to better understand risk for disease. Now, a second study was done by Dr. Sarah Tishkoff and her group in 2019, where they continue to explore the genome-wide association catalog. And it is improving. It is definitely improving. With regards to the number of studies, there's more diversity, and with regards to the individuals. But if you look here at these pie charts, with regards to the individuals, still very few underrepresented populations in the United States are part of these studies. So when we think about the missing knowledge and information to help us to understand health and disease, help us to understand the genetic basis of disease, we do clearly have a situation today where inequities continue to exist. Our institute, the National Human Genome Research Institute, which is one of the 27 institutes and centers at the NIH, is focused on breaking down barriers that impede progress in genomics. This is an important area for us, and we're conducting a number of research initiatives, research consortia, to focus on increasing diversity within the study populations. We now have initiatives that require the investigators to seek out and to work with diverse communities across our country. And it's important in that working together that they work in partnership with those communities. These are 3 examples of initiatives that our institute funds that are focused on increasing diversity with regards to the researchers and the participants in the studies. So now let me take my last few minutes and talk directly about the concepts of race and genetics. And I used this slide that was developed for a couple years ago for another talk that I was giving around direct-to-consumer genetic testing. And this question of why am I Irish yesterday and Italian today? Understanding how genetic information gives us information about our identity and our ancestral background, but also does not give us information about our social identity and how that data is based on the databases that are analyzed versus an understanding of the social experiences and the culture of an individual. So when we think about race and ethnicity, they are complex and fluid constructs. And this is a quote from a paper that Dr. Francis Collins, the former director of the National Human Genome Research Institute, published in 2004, just after the completion of the Human Genome Project. And he said, first, it is essential to point out the race and ethnicity are terms without generally agreed upon definitions. Both terms carry complex connotations that reflect culture, history, socioeconomics, and political status, as well as a variably important connection to ancestral geographic origins. This issue of race and ethnicity and genetics is not a new issue. It's historically has been a major issue and has truly been part of what I would consider some of the challenges that the field of genetics have had historically related to eugenics and scientific racism. And that we must recognize that in 2022 as we move forward to make the field truly equitable with regards to the benefits. So I want to share this, this figure that was developed by Dr. Wendy Roth and some of her work, a professor at University of Pennsylvania. And she talks about the multiple dimensions of race. First, there's perceived race, race that others believe you to be. Reflected race, race you believe others assume you to be. And then racial identity, race you identify as. So thinking about this construct of race, it has various perspectives, how you are seen by others, how you perceive yourself, how you understand your place in the world, and what does that mean? But how does that connect with genetics, and how do we understand that? Well, my research group, as stated, is focused on studying and understanding these issues. And we've been studying these issues from a variety of perspectives. And I want to just share with you in this slide some recent work that my group led. One of my fellows, who's now a graduate student at University of Princeton, led this study that my colleagues at the National Library of Medicine and my colleague, Dr. Lawrence Brody at the National Human Genome Research Institute participated in, where we studied over a 70-year period of time, the American Journal of Human Genetics. So think about this. We looked at this journal over 70 years and looked at how populations were described in the journal. And when was the word ancestry used? When was the word ethnicity used? And when was the word race? And what we found in our study that was published in December of last year, that the concept and the word ancestry is increasing in use in genetic studies published in this journal. Ethnicity is increasing in the use of publications in this journal, but race is decreasing. And what the message that this shares for me is that these constructs are fluid. They change over time. They change based on social context of how people think about these constructs, and they truly are not discrete biological groups. So as we move forward to understand human genetic variation and to understand and change how we think about these issues, we must think about the appropriateness in the use of race. And this work has been discussed by a number of scientists, including the scientists here, Dr. Tishkov and Dr. Roberts. And they say, we believe the use of biological concepts of race in human genetics research, so disputed and so mired in confusion, is problematic at best and harmful at worst. It's time for biologists to find a better way. Well, I just want to share with you that the National Institutes of Health have now sponsored a study that the National Academy of Sciences is conducting. 14 institutes, centers, and offices at NIH have funded this study. It's a study to review existing methodologies, benefits, and challenges in the use of race and ethnicity and other population descriptors in genomics research. And this consensus committee is now hard at work to help guide the scientific community on how appropriately to use different types of population descriptors, including race, in genetics and genomics research. So I want to just close with this tapestry. And as we think about the future of precision medicine and human genomics, that we're all in this together, working together to actually make a difference, to improve health and reduce the burden of disease. And so with that, I want to say thank you, and I look forward to the Q&A.
Thank you, Mr. Bonham. What a great start to this discussion tonight. This was fantastic. Well, I have the pleasure of introducing our second speaker, Dr. Fatimah Jackson. And Dr. Jackson is a professor in the biology department at Howard University. She received her Ph.D., MA, and BA cum laude with distinction from Cornell University. And she's conducted research on population subculture and health disparities. Dr. Jackson has worked successfully and extensively with contemporary and historic African American populations and has conducted research on substructure in this group and the complex influence on health disparities, particularly cardiovascular diseases. She is a master teacher and has successfully mentored hundreds of African American STEM students, including a Rhodes Scholar. Dr. Jackson has invented two computational approaches to identify human microethnic groups and partition their responses to predict the severity of disease expression. Dr. Jackson was a senior Fulbright Fellow in Egypt and has published extensively, receiving multiple awards for her work, such as the Nick Norgan Award for 2009 Best Article published in the Annals of Human Biology. In 2012, she was the first recipient of the Ernest E. Just Prize in Medical and Public Health Research at the Avery Research Institute, the College of Charleston, and the Medical University of South Carolina, at the University of South Carolina. And she was also coined by Rear Admiral Dr. Helena Michaud with the NIH National Heart, Lung, and Blood Institute and the U.S. Public Health Service. So please welcome Dr. Jackson as our second speaker this evening. Thank you, Dr. Jackson, for being here. I think you're still muted. Like you're muted.
Thank you so much for the opportunity to meet with you this evening. And, you know, following back after Mr. Bonham is quite a challenge, but I'll try to build on some of the strong foundation that he laid and kind of look at race and genetics with an emphasis on the genetic information and some of the population demography information. I'd also like to congratulate Baylor College of Medicine for tracking 16 years of progress in genetics and genomics. That's huge. And as Mr. Bonham said, genetics and genomics are really dramatic these days. There's so much going on. The pace at which our technology is increasing is really quite dramatic. We can actually go in and modify genomes, which we were never able to do before. And things that we didn't even imagine 4 years ago, we're able to do in genetics and genomics. And we're understanding more about the genome and all of the multiple factors that are influencing gene expression patterns and the phenotype. So it's interesting that even in spite of the 16 years of progress, we still have a retention of certain concepts, even in the face of significant conflicts to those concepts, such as the concept of race. And so I'll build on this. And I'd just like to say that if you think of the human genetics and genomics as a tree, then ancestry, what we need to do precision medicine, is really at the trunk and the root of the tree. And then all of these other things, the precision medicine, the epigenomics, the transcriptomics, the genomic entropy, all of these other kinds of studies, including pharmacogenomics, phytochemical coevolution, gene environment interactions, they're up in the foliage. So if you don't have a strong background in the ancestry of the individual or population that you're studying, the tree, all that's up in the foliage will not survive. And that's kind of the situation we find ourselves in. In fact, the persistent bias that Mr. Bonham spoke about is very persistent. So we look at the genetic variability in legacy African Americans, meaning those individuals of predominantly African descent who are derived from the legacy of the transatlantic trade, slave trade, the trade in enslaved individuals, and chattel slavery and Jim Crow racism. So in North America, we are calling these populations legacy African Americans because actually there's a growing diversity in Black America. And so not everyone is coming from that heritage, but the majority of African Americans are legacy African Americans with origins in primarily West and West Central Africa. And in fact, this African diversity is the greatest source of admixture in legacy African Americans. This population also has modest gene flow with non-Africans, and that primarily we're talking about North Atlantic and Iberian Eurasians, Western Eurasians, meaning Europeans, Eastern and Southeastern Native Americans or First Peoples, and that's at least in North America. When you move down to the Caribbean and to the legacy African Americans in South America, the pattern of admixture with Europeans and with Native peoples varies. We also find in these legacy African Americans in North America significant non-genetic sources of variability by their geospatial region, and that's where the substructure comes in. And we can find some regional variation, which is quite interesting. Now, this is what the persistent inequities or persistent bias that Mr. Bonham spoke about. This is what it looks like genomically. This is the chromosome profile of an African-American, US-born African-American woman. But you'll find that there are huge sections that are unassigned. In fact, 64.2% of her genome is unassigned. The red areas represent West African heritage. The blue areas represent European heritage. And in fact, we know more about her European ancestry than we do about her African ancestry. And this huge number of unassigned it's unassigned to any geographical region, or that means that there's no data currently available. Now, this is against the backdrop of Africa, the continent of Africa, as the ancestral homeland for our entire species. So our species, Homo sapiens, has spent the bulk of our existence in Africa, which means that there was ample time for the accumulation of mutations that are at the foundation of the genetic variation that we see. And there's now, we understand that there's more variation in African descended individuals than in any other human population. And it's because of this long evolutionary record. But as Mr. Bonham pointed out with the testing, it's the population that is among those we know the least about in terms of their genetic variability. Now, when we look at the substructure in legacy African Americans, we see that really African Americans are a subset of sub-Saharan Africans. And this is a diagram from one of Dr. Tishkoff's papers. And you can see the African Americans are here in purple, and they are simply a subset of the orange colored sub-Saharan Africans. And you can see that we very closely are tucked in underneath that population, and even closer to the sub-Saharan Africans than we are to the Cape Town admixed populations. Now, there is a great deal of complexity in the indigenous African origins. As I mentioned, humankind originated in Africa, spent the bulk of our time in Africa. Africa is the most tropical of continents. And we know that Dobzhansky, a great leader in evolutionary biology, reminded us that in the tropics, the rate of evolution is actually faster. So you get an accumulation of genetic change across Africa that we can see when we use this as a structure program. It's a computational program that allows us to discern the patterns of variation across geographical regions. And so when we compare Northern Africa, Western Africa, Southern Africa, and then African Americans, you can see that African Americans bear strong resemblance to Western Africa, but not solely that, because we also have gene flow from other parts of Africa. This is the middle part of Africa in here, and then Eastern Africa here. So within the continent of Africa, there's not genetic homogeneity. There is heterogeneity, great heterogeneity, and that is becoming increasingly reflected in the growing diversity of Black America, because people are coming now to the United States from all over the continent of Africa and from throughout the transatlantic diaspora. This was a diagram that I just picked up off a Black demographics page, but it gives you an idea of the export regions from which many African American ancestors came, and then they were brought to particular areas on the East Coast of the United States, and then the southern, southeastern part of the United States. What we're finding is, although these are the traditional maps, what we're finding is that these points of origin actually go deeper into the continent. So a place like right here, which is Burkina Faso, ends up being extremely important and pulls people from Mali and from Chad and so forth, and then they end up exiting Africa at the Bight of Biafra or the Bight of Benin. But they originally are coming from the Sahel region or from the tropical rainforest regions. This is a little more data, and these pie charts are my own data, where we've looked at the movement of Africans to various staging areas in the United States. So the Chesapeake Bay area received 38% of their Africans from the Bight of Bonny. This is also known as the Bight of Biafra. The Carolina Coast region, the Bight of Bonny was much smaller at only 7%, but 40% came from West Central Africa, which is really quite amazing because, and these were intentional patterns of securing enslaved Africans, because the Africans that were brought to the Americas had skills. So rice cultivation was extremely important in Carolina. West Central Africa is a big rice growing area. Mississippi Delta region is kind of interesting because West Central Africa was only 25%, Bight of Bonny only 8%, but look, Senegal, Gambia, 32%. So the Mississippi Delta had a different pattern of African input into that region than, say, Chesapeake Bay. And it's because of the inherent diversity in Africa, we think that that diversity in Africa is reflected in the variable pattern of health disparities across African American landscapes in North America today. This diagram, this table rather, came from tracing African roots, and it's just their numbers of the proportion and numbers of Africans brought to particular regions, from particular regions in Africa to particular regions in North America. Now, when some of the researchers have looked at legacy African Americans, they've kind of rejected the idea of regional variability. And they produce, like, this is structure again, and you don't see significant regional variability from Chicago to Pittsburgh to Baltimore to North Carolina. But I kind of take issue with this. And in fact, what we're understanding as we learn more about the genome is that it's not simply these alleles that they looked for that may be expected to differ. In fact, we may see differences in the entire structure of the genome, with certain areas of the genome occurring in particular African-derived populations and not in others. We also know that the epigenome, those chemical markers that are in association with the genes and can influence gene expression patterns, that the epigenome may also vary from Chicago to Pittsburgh to Baltimore to North Carolina. Also, by the way, a lot of the people in Baltimore actually came from North Carolina. So without history, you end up not having a very good research design. You got to know the people's history. And of course, Chicago, a lot of the people from Chicago came up from 5 counties in Mississippi. So if you understand the pattern of migration, then you can explain some of the similarities or lack of substructure that seem to appear. But we have hidden complexity. It's in the microbiome, which are the microorganisms in our body that have a tremendous influence on our disease profiles. And it's in the complexity of gene-gene and gene-environment interactions. And so one of the things that we've needed to do is study each of these regions where African Americans have been living and really characterize them in terms of their genomic diversity, their epigenomic patterns, the microbiome patterns, because the epigenome and the microbiome are as important as the genome in disease expression and genomic expression. And then we need to understand the environment rather than assuming that the environment in all of these regions is identical. So this is some of the diversity from Africa. I just picked this slide up for a project that began in Novaris looking at African genetic diversity. And you see differences in head shape, differences in facial expressions and so forth, all within continental Africa. So I'd like to summarize by giving you an example of why the genetics is so important in understanding the foundations for precision medicine and comprehensively addressing the health disparities that emerge. One big health disparity that is quite evident among many African Americans is end-stage renal disease. And the identification of risk alleles in legacy African Americans is often correlated with their enhanced risk for specific chronic diseases like end-stage renal disease or chronic kidney disease. So here is a result that we got from a patient where this patient had, she was doubly heterozygous for 2 risk alleles for chronic kidney disease. And these were the diagnostic findings when a genome-wide association study was done. And not only does the genetic data provide enhanced diagnostic information, the genomic data could also be the basis for preemptive interventions that through gene environment and gene-gene interactions can dramatically reduce the risk of ever developing the associated chronic disease. So the genetics doesn't cause disease, it sets up the risk, it increases the risk. If we can control the environmental triggers for those diseases, we can have a huge impact on the expression of the disease. And this is just a diagram showing the apolipoprotein L1 gene, the ApoL1, and the renal risk variant. And this is one that affects the kidney, it affects the liver, it affects the heart. And the triggers are numerous. When I look back at the gene and saw for this doubly heterozygous individual, she was, without intervention, she was going to get end-stage renal disease. And what were the triggers? Diabetes, hypertension, general psychological stress, excessive psychological stress. So all of these things worked in together. It helped us predict. And this is what precision medicine can do if we have the ancestral background on the population. This particular APOL1 gene, the risk genes that we found in this individual are very common in Central Africa, West Central Africa. And in that setting, they seem to provide protection against trypanosomiasis, sleeping sickness. But in Chicago, very little sleeping sickness in Chicago, very little trypanosomiasis. So the gene that was adaptive in one setting ends up being maladaptive in a different environmental setting, kind of like Mr. Bonham's work with sickle cell, where it's adaptive in one setting and less than adaptive or almost maladaptive in another setting. What do we do about it? Well, that's the next step. But at least being able to connect the ancestral foundations and build that up so that we can do precision medicine and increase our abilities at pattern recognition, increase our abilities at detection. Then we can begin interventions that are initiated in infancy rather than waiting for the disease to express itself. So anyway, that's what I had to share with you today, and I thank you all very much. And I'm looking forward, as Mr. Bonham said, to the Q&A, because there's so much to talk about. Thank you.
Thank you so much, Dr. Jackson. There is a ton to talk about, so thank you very much for your presentation.
Sure.
And now I have the pleasure of introducing our third speaker. And with this Evenings with Genetics seminar series, we always try to include family voices. And so it really is an honor tonight to include Dr. Cheryl Shatting. She professionally is a passionate expert and advocate for training, mentoring, and developing students of color in the biomedical sciences. Personally, she is a single mother and her son's biggest fan and advocate for him to receive the necessary services to help him thrive and succeed in the school system and in life. She enjoys quality time with her son, from playing in the park to container gardening to introducing him to music from the 80s. So please give a warm welcome for Dr. Shadding.
Good evening, everyone, and thank you all for this opportunity. And this is certainly a hard act to follow. Thank you, Dr. Jackson and Mr. Bonham, for wonderful, wonderful talks, and to Susan and Dr. Murray for inviting me. So this is a different hat for me because I typically don't speak publicly about my son. I'm usually on the other side talking professionally. But today I'm just a private citizen and talking as a mom. So, I just wanted to share with you all that, I do have a son. He's 8 years old. He'll be 9 next month, as he keeps reminding me. But when he was 3 years old, he was diagnosed with mild to moderate autism. And I would have totally missed the diagnosis and missed the signs because I thought he was just being a typical boy. He was very rambunctious. He was very social and still is, and loves to talk, loves to meet people, and very chatty. And I just thought all those things were normal, and all the signs that I knew of with autism were the opposite of that, were people who were more distant and more withdrawn, and that was not my kid at all. So I was very confused initially, but the signs came through him going to daycare and talking to teachers. And so I would drop him off, and I need to add the backdrop, since this is about genetics and Black History Month and talking about equities or inequities. Typically, he's like one of a few Black children in a place. So he was going to a daycare where he was one of literally a handful of Black children. So when the teacher would tell me things, I'm thinking, well, he's a Black kid, so you're just not used to this, and he's being a boy, and that's it. And I, just kind of passed it off. And then I changed daycare centers to one that was very specialized in early childhood development because a spot opened up. And they did assessments through their local school district. and they come to your home and tell you about the assessments and go over them with you. And at the time I was married, and so I thought, okay, they're going to come and they're going to tell us that our kid is a genius because he's so smart. He learned to read very early. He knew his alphabet. I know that's what they're going to tell me. So they sat down, they gave us the test, the results of the test. And, you know, I'm looking at all these low numbers and I'm thinking, is this like golf where low numbers is a good thing? But that wasn't true. And so I was very disheartened when they were telling me, you know, they couldn't diagnose him, but they were telling me that they noticed various delays, social delays, language delays, and I just couldn't get it. And then they referred me to my own school district for further assessment. So we did that. And they just kept telling me, you know, the same thing, but that they couldn't make a medical diagnosis, that theirs was only an educational evaluation. And the school district that he was in at that time, I felt like the person was telling me like a little secret saying, I cannot give you a diagnosis, but this is what this is saying. And he can have an IEP in school and giving me all these resources. She's like, but you need a diagnosis. I said, oh, so I need to go to his pediatrician. So I went that route, went to the pediatrician, got him into a neurologist. And the neurologist officially diagnosed him after spending like 2 hours with him and watching him and observing him play and talking to him and all those things. But when we sat down with the neurologist and, you know, he gave us the diagnosis, I think I was just completely numb and just didn't really understand what this meant for him and what I needed to do as a parent. So I sat on that information for a while. I didn't do anything. He gave me a big binder of information. I did nothing with it because I didn't want to accept it at the time. But I got to work. I read through the binder and started reaching out to people. And that's how I learned how to get him the resources that he needed, either from the state, from his school, and privately. Fortunately, he was able to get different services through school because he did have an IEP. Which is an individualized educational plan. I personally had issues with it initially, but it depends on how things work out for you and how you kind of use that system. I was very cognizant of the fact that he is a Black male child, and I didn't want him to have all these labels on him. But at the same time, I knew that the things that he was getting could be very helpful to him in the long run. And those things did help him. He was doing education therapy at school, and I was able to get him private therapy as well. So he was kind of getting a 1-2 punch, and he really thrived and really did well until we moved to Houston, where he didn't have all those services anymore, which, you know, maybe I'll get questions because I'm running out of my time about that. So moving from where we were to here, actually, there's been a huge difference. And I've even seen some decline in the progress that he made over time. But you do have to be an advocate for your children. And I even have, well, all of his teachers pretty much tell me, wow, we usually don't get parents that are this involved and ask us these many questions. So you have to be that parent in order to get the information and the services that your child needs. But you have to first accept that there could be something going on, that it's not necessarily a label, but even if it is a label, it can help your child get to a different level than where they are. And we found that it was just very useful for him to have the different services that he had to succeed in the classroom. All this time, he's always been in what's referred to as a general education classroom. So he was never, quote unquote, special education. I insisted that he will always be in a general education classroom with just is it called push out, pull in services. So, so you have to have that voice and let people know that this is your intent and this is what you want to happen. Because I was always have been very vocal in his IEP meetings. I would call the meetings, and that would make them nervous. But it's like, well, I need to talk about this because I'm concerned about what's on this list, and I've noticed this, so let's get this going. So I would just encourage parents of people that you know to just be very vocal about what's going on with your children. And I'll stop here and I will take any questions when that time comes.
Thank you so much, Dr. Shadding, for talking about your journey and working with your son and all of the services that he needs and that I hope he is able to get now in our lovely city. So yes, I guess would we like to open it up for discussion with the speakers, and Dr. Murray is going to lead us from here.
All right. I'll start it off. I have like several different ways I would love to go. I wish we had all night, but to you both, I'll try to make it as big as possible. So what do you think it would take? How long do you think it would take for us to realize the promise of precision medicine, given that we have missing data, cell lines that aren't exactly what they were supposed to be? You know, how long would it take for us in a perfect world to get there? What are some of the most important factors you think need to be in play for precision medicine to be the promise for all?
So I'll start. But first, Dr. Shadding, I wanted just to acknowledge your comments and make the comment that as a parent and as a caregiver, your role is so important with regards to benefiting your child, but also being an advocate for your child. And as we think about health inequities in our country, we must think about this question about sometimes people don't get the services they should get. And it's not that they're not there and not eligible for them, it's that they need to make sure that they have someone advocating for them. And so I guess that's a message to all the families out there, whether it's related to a genetic condition or not, that's so important. But to your question about how long will it take us? So, you know, this is such an exciting time, I think, in science. I mean, you know, I've been at the National Human Genome Research Institute now for 19 years. So I arrived just before the completion of the Human Genome Project. And that was the project to sequence the first genome. And you think about the millions of genomes that have now been sequenced across this country and across the world. All the new knowledge, how quickly we are learning new information, our ability to analyze data. So I am hopeful, okay, if, and I put this if out there, if we truly focus on that we must have diversity in study participants, and we must have diversity in the researchers who are doing the research. And people thinking about the questions from different lenses, different life experiences is required for us to move forward. So I think we have a chance to truly have diversity, to work with communities across this country, across the world, to engage them. But we need to make sure that the scientists that are involved also reflect what that community looks like.
Absolutely. I completely agree. We have to have diversity. We have to have the science is proceeding so quickly that even over the course of 2 or 3 years, you're able to do things that we just never imagined. So we need to train. I mean, to me, it's an issue, a national emergency. We need to train students of color so that they will be prepared to go in these labs and to do the research. It's very hopeful that on the African continent, we have a number of entrepreneurs and businesses. And of course, NIH is also supporting a big genomics project, the H3 initiative. But we need more indigenous involvement, whether we're talking about African Americans, legacy African Americans, or continental Africans. Everybody is mostly concerned about themselves. That's why we have this disparity in genomic studies, because the researchers have studied the populations they have easiest access to. People like Sarah Tishkoff are an exception. She's not from the African community, but she's devoted her life to studying African genomic diversity. But that's an exception. In fact, I can probably count on one hand the people around the world who are not African, who are interested in African diversity. But that's got to change for a whole host of reasons. And, you know, we don't have a choice. We can't wait for someone to recognize us. We have to get out there and do it ourselves, because it is, if we don't get the ancestral information, if we don't understand the gene environment interactions, if we don't have a more sophisticated view of ourselves, not only will we fail to get the precision medicine that we need, we'll fail on so many different levels, because the train is moving and we're not on board. We need to get on board. Otherwise, we're going to be run over by the train. And we'll be talking about those, remember those legacy African Americans that used to exist? Yeah, they all died out of sickle cell and end-stage renal disease. Yes, shame on us. So we've got to do better. We've got to be actively involved. And just as you are doing, Dr. Shadding, with your son, we have to do that for our people to push. Where is our representation? You know, we really have to do this. Diversity is, we say freedom is not free. When diversity doesn't just happen. They say when a bad idea like racism stalls in a population and is persistent generation after generation, it's serving some other purpose. Everybody knows it's stupid. Everybody knows it's a dumb thing to classify people by race. No biologist worth her salt would say, oh yeah, races really exist. Maybe in sparrows, but not in people. We're one genus, one species, but we have allowed the social environment to dictate the science, and we can change the social environment. We can make Black Lives Matter.
All right, thank you. Kareem Watson, who is the NIH All of Us research community engagement, the national community engagement director, ask a question. He says, this was amazing. What role does community outreach and engagement play in ensuring the diverse populations have access and awareness to precision medicine research? So, and you've touched on it a little bit, but you can take it a little further.
So I'll just start just this time, and then I'll turn it over to Mr. Bonham, because he understands even better than I the community part. But the community, do you remember when Black people were afraid to have genetic testing, that they said, oh, don't test me for sickle cell, it might give me sickle cell. You know, and even at the beginning of the Human Genome Project, people were, I don't know about that, they were hesitant. Or it was beyond genetic hesitancy. It was genetic rejection. They did not want to be tested. They thought that even talking about, you know, the potential to find disease or risk alleles might cause them to have the disease. So we need to educate our communities so that they can be active participants, you know. And it's not just this, it's in clinical trials, you know, where our people have not been present in the clinical trials. And yet we want the benefits, but we're not willing to pay the cost. And the cost is engagement. And that's what, to me, community involvement, community engagement is going to be essential, because the handful of African-American scientists and administrators and are just not enough to carry the momentum. We really need to have a grassroots support for massive ancestry testing. I think that ancestry testing should be a part of reparations. If people are talking about reparations, really, tell me where I'm coming from. Tell me who my people are. If you tell me that, I don't need your $1 or your $2 because I'm just going to give it right back to you, when I buy another Tesla or something like that. So tell me what I need to know to have a better quality life. And that's the kind of demand I think that we in the community, the African-American community, need to be making of all of our so-called officials, I don't know. This is the preacher in me coming out.
Yeah, I think you're in the pulpit. So I would like to just add to that and to Dr. Watson's question is without communities' involvement, we will not be able to make sure that the information that is being learned actually benefits the communities.
Yes.
And without the communities, we will not be able to transfer that information to the communities.
Mm-hmm.
And I'm just going to give a nod out to the All of Us program. I think that All of Us program, when you look at a model of programs, is an example where there's truly that work is going on. I'm sure there's a lot of things that they want to continue to improve upon, but that engagement on the ground with people in a community, diverse communities across our country is so important. And coming not to say to do this, we're going to tell you what to do, but to listen to the communities and to listen to what is important to the communities with regards to their health and their needs. So it's fundamental to the future of the advancements with regards to genomics and genetics, but it's also fundamental to all of the sciences of this engagement with communities with regards to how do we work with communities to help to advance research and advance science to improve health.
Yes, yes.
Good. Thank you. Wonderful. And another shout out to the All of Us research program. We too have a grant and an engagement grant where we are engaging the underrepresented biomedical researchers with the All of Us data. And we'll have a conference. It's in May. So please go to the Department of Molecular Human Genetics website and find that information. So next question, Pooja Rani says, thank you, Dr. Jackson. Can we apply computational biological, must be biology, to find the complexity of gene-gene and gene-environment interaction?
Yes, yes. In fact, we have to, because the data is too complex for our little brains. We have to use computers, and computational biology is extremely important. And one of the companies that I work with, QuadGrid, is a data science company. And one of the things that company, it's a minority-owned company. And one of the things that we've been doing is developing algorithms to reconnect Africans from the transatlantic diaspora, African Americans, with their multiple points of origin in Africa. And not just where they exited Africa, but the empires and kingdoms that their ancestors originally came from. So I'm really excited about this because it's the application of computational sciences to a historical issue that is of important psychological value for many African Americans. But it's getting at that problem of lack of ancestral information and assumption of genetic homogeneity across all of African America. And those are two problems that impede the development of precision medicine, So yes, so absolutely, we're trying to use the computational approaches, trying to get into big data. And of course, NIH is the repository of the biggest databases. So at some point, we'll be coming to you, Mr. Bonham, and saying, hey, we need some help here. But we're trying to develop this algorithm that will allow us to make predictions. If we know you came from Baltimore and your people arrived in 1724, then we can find which ships came into the Baltimore harbor. We can track them back to particular places in Africa. Then we can use the genetics that you're manifesting to see, are there links? So far, we found one family that is an African-American family that shares genetic heritage, explicit genetic heritage, with a family in Benin. And they allowed me to sit in on the call when they contacted the people in Benin. And it blew my mind, because they were looking at the people in Benin and the people in Benin, this is on FaceTime, were looking at the African Americans, and people were waving, and it was like 400 years was traversed. And it felt so satisfying, because the bond that was attempted to be broken, we put it back together with genetics. And we need to do this over and over again, because that's the salvation. So community engagement is on both sides of the Atlantic and throughout the Americas.
Oh my goodness, that would be awesome if everybody could have that experience.
I
Oh my-
know. I mean, we were all kind of hanging on to Alex Haley's roots, but now we can do better than that because of the genetics, you know?
Right. Oh my goodness. So, well, I won't go there. Next question. So Lori Banks, actually it's Dr. Lori Banks. Up and coming students, undergrads in the biosciences seem to have little understanding of health disparities. Are there updated comprehensive resources available to fix that in the classroom?
There are a lot of great resources out there, and I'm happy to follow up with her and giving her some specific resources. But I just encourage you to look at some of the things that actually the AMA is doing, work that Robert Wood Johnson Foundation is doing, things that are coming out of the National Institutes of Health. I think that there's all kinds of opportunities to provide resources for students to help to expose them around issues of health equity, health disparity, to understand and have conversations, challenging conversations, difficult conversations of why we have some of the disparities that we see in our country.
Mhm.
Okay. Another question from Dr. Astra Sparks. I know these people. She said, what role does the pharmaceutical industry play in the diversity in genomics?
So you're asking about pharmacogenomics, because pharmacogenomics is extremely important. It's one of those areas of study that's up in the foliage. But again, you have to have the ancestral background. I'll tell you that I've been at Howard 8 years, and maybe about my 5th year in, the dean of the medical school said, I want the medical school to have the same kind of treatment that Jimmy Carter got. Because former President Carter suffered from cancer and then received precision medicine, so he is in remission. Well, I said to him, yeah, but we don't have the baseline information yet. We don't have adequate baseline information to build that kind of an intervention. And he was disappointed, but the reality is that without the baseline, we can't do much. We're testing, we're shooting in the dark. And it's true with the pharmacogenomics, it's true with the epigenomics, it's true with all of those things that were up in the foliage, that the ancestral base, the ancestral foundation lets us know who we are dealing with. And then we can design our drugs based upon that knowledge.
I would just add that there are a number of companies that recognize this major gap in our knowledge. And the gap in the knowledge is not just for those communities that have been left out, but it's for everyone. Yes. to understand the variation. And so So there is, active efforts going on across the globe to really to increase the diversity in studies of recognition of these issues. And I want to applaud the companies that are really moving forward, but there's much more work to do for all of us to do.
Mhm, mhm.
Okay. Question from Nadia Merchant. How do we get minorities or underrepresented individuals to get through clinical trials and testing in the most effective way? I know it's easier if I can connect two families, but how can we do it as providers, specifically African Americans, since they are a minority in trials, unless it's targeting them?
Yeah. Hmm.
I'll start on that one, and I actually want to bring Dr. Shadding in. So the question of participation in trials. One of the first steps is for those primary care providers, those specialists, those physicians that are involved in your care to make you aware that there are trials that are out there, that there may be trials at your institution, or there may be trials at another institution that may be important for your child or for yourself to be part of. It's important for all communities to learn about the variety of different types of clinical trials that exist. And so I think there's a responsibility on physicians and other healthcare providers to make sure that as part of their treatment plans, they make the family, the patients aware that clinical trials do exist and that a clinical trial may be of value to that individual or a value to others with the same disease and condition.
Yeah.
And so it's a two-way street with regards to engagement around clinical trials.
Yes. Yes, I agree. Those are very important points. Very important. Yeah.
And I would agree. So from both sides, as a scientist, but also as an advocate for a parent, and I was a caregiver for my parents, and also actually my ex-husband is a cancer survivor, and when he was going through that, they there were people like in the waiting room that were trying to sign up patients for clinical trials and for trials that the physicians were doing, and probably the same thing for my parents. So there has to be education on the side of the patient. They have to know what they're getting into or signing up for, but also the medical personnel have to provide enough information with the informed consent and educating people, excuse me, to let them know what all will happen, because there is still some distrust and still some
Yes.
understanding on the part of the patient. So sometimes when I see things or when I receive consent, especially for my son, gosh, I've even had to tell people, I'm like, I'm not signing this because this doesn't give me enough information, and you shouldn't have this question here. And so anyway, I won't go into all of that, but there has to be enough information provided to the people that are signing up. And it is good for the patient to have people in their corner, whether it's an advocate, whether someone that can explain those things to them. So I definitely agree. It's a two-way street, but I've certainly have seen holds sometimes, especially on the educational end, when it comes to truly informed consent.
Mhm, mhm. I think trust is a huge issue. And when you look at the history mediates against trust. So why should I trust you when I just remembered, oh, all of this litany of distrustful experiments that you all have been doing on my people for 400 years. So what we've got to do is build the trust and informing people, opening up the diversity of practitioners so that people see themselves in the person asking them to participate and assuring them, although there's no guarantee, we have Tuskegee to look at, there's no guarantee, but the trust, at least you begin building the trust.
Yeah, that's true. And I think everyone should be involved, because when a person goes into an environment, the way they are treated by all those individuals
Mhm.
adds up to whether they will trust or distrust that place.
Right.
And then someone comes and asks them to participate in a study, then the answer is probably no, because how are you going to treat me when I walk in and you ignore me? Or
Exactly.
yeah.
Mhm.
So it's more involved than I think people want to realize too.
Mhm.
I'll bring the last question from Helen Hernandez. I saw at one point that the same treatment for visceral leishmaniasis had drastically different outcomes in people in Asia than it did in Africa. How do you decouple genomic, pharmacogenomic factors that might lead to that versus societal, environmental, or any number of other factors?
I think we have to do reverse genomics on differential clinical responses to leishmaniasis. I mean, because you get a different phenotype. So then you go back and then try to figure out, okay, what are the genes that are involved in producing that phenotype? So we're starting with the end product and then kind of reverse engineer it to figure out what are the genes. And usually to do that, you have to mutate the gene or mutate the candidate genes or candidate segment of the genome, or you can do a knockout model. And either one of those approaches, either mutagenesis, intentional mutagenesis, or a knockout model, could allow you to use reverse genetics to characterize what is it in the genomes or the environments of Asian people that give them a differential phenotype in response to the same infective agent compared to Africans. I mean, but I just got through teaching about this. That's why I'm thinking about reverse genetics. I don't just walk around like this. This is because I was teaching about it today and I was like, okay, I'm understanding this. Let me explain it. And when you said it, I thought, there it is. Then with our tools now in genetic engineering, we're better able to tease apart, to deconstruct all of the factors that produce that particular phenotype. We don't have to do what's the old way is forward genetics, but we can do reverse genetics and actually identify which segments of the genome are most important, if there is a segment that's important in producing the differential phenotypes.
Thank you all so much. I mean, this time has just whipped by really fast, and I hope you will consider being presenters again in the future, because I think we could easily spend a lot of time with you. Thank you very, very much, Mr. Bonham, Dr. Jackson, Dr. Shadding. This was really remarkable. And I just think that, thinking about the future with STEM for underrepresented students, if they could hear presentations like this, I think they would be on fire. So I thank you very, very much. And we will continue all of our efforts to engage the community in all of this genomic care and moving forward. We thank each of the attendees for being here and welcome you to answer the survey that'll help us plan our next webinars. The March 8th topic will be on 21st century treatments for genetic diseases. So I thank you again for an absolutely amazing webinar. And thank you, Dr. Murray. This was fantastic. I wish you all a good night.
Thank you. Good night.
Thank you so much. Good night.
Good night.